Now I will introduce the chair of the next session, Professor Bai O. She is the Deputy Director of Hematology, the first Bethune Hospital of Jilin University. She's the head of Lymphoma Special Disease Alliance. And welcome, Professor Bai O.
Hello, everyone, and every professor online and local. I'm glad to introduce Dr. Yi, and Associate Chief Physician, and he came from the Institute Hematology and Blood Disease Hospital of Tianjin, and he is minor members of following. But the major one is that committee of lymphocyte disease group of the China Society of Hematology and Chinese Medical Association. And he visiting and learning in America for 2 times and published more than 20 papers, and also get the foundation support from the Chinese government. And the topic he will give us the biological characteristic of China peoples. Welcome, Dr. Yi, please. Thank you very much.
Thank you for your introduction, Dr. Professor Pai. And it's a very great honor to be here to present some of our data. And very nice to meet you, Dr. Helmark, Hillman, and Rosie, and Gail, and other professors online. And now I will introduce some of our work on the biological characteristic of chronic leukemia, lymphocytic leukemia in China. As we know, CL is the most common adult leukemia in the Western population, and we see, but it's lower in the Eastern, including China. From this picture, we can see that the incidence is as low as 10% in China, and so we because the basic population and the total population in China is already higher, and it's four times larger than in the US. So the total instance of the cases per year is about 700,000 to 800,000 cases per year in China, I think. But it's still lower than there were maybe 21,000 newly diagnosed CLL in American, I think, in the 2020 years. So the lower instance of the CLL in China maybe indicate that there may be some different biological background. And we have also described our patient, and we found that in totally there was 2,000 patients was diagnosed in our hospital, including 1,300 patients in hospital and 700 patients outpatient. And from this timeline, we can see that the total number of patients was increased along with the time. So that's maybe also indicated that the incidence of CLL in China may be also increased with the years. The middle age of this population is about 16 years old, and that's very close with as Dr. Xu said, this is 59 years old. So the middle age in Chinese population is younger than that in the Western population. And about the real stage and the benign stage, we can see that there's only 10% patient at the real 0 stage, and the benign stage is also only 13%. And I think this percentage is in the Western population is about 16 to 70%. So most of patients may be diagnosed at the very advanced stage in China. as we know, the biological characteristic has been described very largely, deeply in the Western population. In the last ASH meeting, and there was many dramatic progress of this pathology. So, for example, I listed the title of this publication, this oral presentation, in the last ASH meeting, and we also have illustrated how these genes work in the initial or in the progress of this CLL disease. And I think the main biological characteristic of CLL is about 3 aspects. One of them is cytogenetic aberration. Another is very important about the IgHV mutation stage, and others is the somatic genetic mutations. And as we know, 20 years ago, the physician has discussed that the mutated cell and the unmutated cell has dramatically different clinical outcome. And until last year, our scientist has answered the question. Using the sequencing, we can divide these two populations enriched in different gene mutation, driver mutation genes, mutated genes, and copy number variations. So, although there's some overlap, but most of them are enriched in one of the groups. So our study is also focused on these three aspects: cytogenetic aberration, IgHV mutation, and dramatic somatic mutations. In respect of the cytogenetic aberrations, and our group and the Dr. Jianyong Li's groups, and also other groups in China, and has described the frequency of mutated genes. I list here is our cohort and Lanjin's cohort, and the instance of deletion 13q, trisomy 12, deletion 11q, and the deletion 7p, and translocation Ig40q. I think the instance is comparable between the Western and the Eastern population, a little bit higher in the deletion 7p and the translocation of IgH. Maybe there's some association with the advanced stage of this Chinese population. In respect the IgHV mutation, as we know, this is the basic biological characteristic of CLL. Previously we have collaborated with Dr. Robin Fo and analyzed the IgHV mutation, and we compared the data from their group. We can see that the instance of the IgHV mutation mutated groups is about half and half in the Western reports, but there is more people characterize the mutated IgHV. Near 1/2 of the patient has the mutated IgHV genes. In about the fragment usage, I think there's also some difference. For example, the fragment usage of V327 and 439, and also some other fragments usage is higher in the Chinese population. But the most frequent usage fragment about 1 to 69 is very low in this population, in our population. So that means the Chinese population has some distinct IgHV mutation and usage. And then we also increase our cases and compare it with the Europe and the USA data, and we got some similar result, but a little bit difference, because when we increase our cohort size, we found that the fragment of 4 to 34 is also higher in our population. About the stereotype, I think this is also very important for this CLL biologic. And the substate 2, subset 1 is very frequently used, happened in the Western report. But in our cohort, we found that the subset 8 and subset 4 is the major subset in our cohort. So in total, we found that there was a much higher percentage of the mutated IGHV in Chinese CL, and also lower usage of fragment usage 1 to 69, 1 to 2, and 3 to 21, but a higher frequency usage of 3 to 7 and 4 to 13, 4 to 39, and there's some higher usage of the subset 8 and subset 4, but lower for subset 2. Next, we would like to take a look at the somatic mutation landscape. As we know, in the Western population, we have reported there were 1,100 projects, and the most frequently mutated genes is about S3B1, Notch1, ATM, BR3, and TP53. But in our group, we have sequencing 300 patients in using the target sequencing next generation sequencing technique, and we found that the S3B1 mutation rate is lower in our population, but the instance of MID18 mutation, Mutated mutation, is super higher in our cohort. There's maybe 30% patient has MID18 mutation, and 8% of patient has the KMT2D mutation. And the mutated genes can enriched in the chromatin modifier, the BCRs in pathway and DNA damage, MOC pathway, Notch signaling pathway, and RNA metabolism pathways. Compared with the newly diagnosed patient, the relapsed patient has a higher instance of TP53 mutation, ATM mutation, and the IGHV unmutated patient, the complex kilotype, and also deletion 7q and 7p. But the MID8 mutated genes was not found in this relapsed patient. And the mutation TP53, ATM, KMT2D, and Notch1 also associated with the poor survival, both of the peer progress, time to the treatment, and the overall survival in our cohort. And this mutated genes also associated with shorter time to the first treatment. So the KMT2D is also an independent factor for this time to the first treatment. So that means that KMT2D may be a functional mutation. Now, although there was high instance of this KMT2D and MID18 mutation in our cohort, but we still cannot say that this is the unique mutated genes. Then we analyze the relationship between these two mutated genes and the IGHV mutation and the usage. We found that the KMT2D mutated genes was higher in this fragment of 4 to 34 and 4 to 39, and also a little bit high in the 3 to 7 fragment usage. So as we said that this usage is also highly happened in Chinese population. So the situation is also in the MID18 mutation. As we say that Chinese population has a higher mutated IGHV, so the MID mutated genes is also happening in this population, and happen in this fragment usage 3 to 7 and fragment 4 to 34. So that means there was some population, some patient carried the mutated the distinct usage of fragment IGHV fragment, and also have the very high mutated KMT2D genes and MID18 genes. So that means this population maybe has some distinct biological characteristic and mutated genes. Then we compared the clinical features between the MID8 and mutated genes, and we found that MID8 mutated genes haven't have more frequency in the male patient, and there is also a higher frequency of monoclonal gamma and the higher HPV infections, including the antigen positive and core antibody positive. And also there are some also associated with some other biological characteristics. For example, as we said, the unmutated stadium is also is lower in this mediated mutated genes, and there are also some association with the lower instance of this deletion in 70p and 11q. And as we said, there's no mediated mutation in this in our relapsed patient. For the KMT2D mutation, as we know, KMT2D is control the methylation of this histone protein 3 K4 site. So if this gene mutated, it will decrease the methylation of this protein. So we have using the immunofluorescence inside hybridization technique to say if the KMT2D mutation impacted this site, the methylation. So this is wild type patient. This is the mutated patient. We can see that mutated patient has a lower expression of this proteins methylation. So this means that KMT2D mutation is a functional mutation. So the KMT2D is a epigenetic genes. Can these genes mutated? We can use some epidemic modifier drugs in this patient. So we have using the primary CL patient cells to compare the sensitivity of with some drugs, for example, the decitabine and the childamide, and also compared with the sensitivity of ibrutinib. We found that the KMT2D mutated genes are more sensitivity to this decitabine and the childamide, but had lower sensitivity to ibrutinib. But if you use these drugs together, we can find that the ibrutinib and decitabine have some synergistic if they work used together. So in summary, there are some distinct characteristics in Chinese CLL, including the IGHV genes usage in mutation, and also there may be, because these two genes will happen more frequently in this distinct IGHV usage population. So that means there's some of the population have the distinct biological characteristic in Chinese CLL patient. Thank you very much.